Test.
Test.
Okay. Okay. Hello, Nikhil. Thank you
Hello.
for joining me.
Thanks for having me here.
Yeah. So, I wanted to ask you, what exactly is Huntington's? Like, if I were to look inside my brain right now and look at Huntington's disease,
hmm
what would I see?
Well, Huntington's disease is a genetic disease where a change in the code of your genes causes the gene to be abnormal, and the resultant protein, which is synthesized from this gene, is faulty, which would eventually kill the neurons. So, Huntington's disease is called a neurodegenerative disease, and the brain area specifically which controls your bodily movements. So, to understand bodily movements, we can look at it in two ways. One is having the ability to do what you want to do, and having the ability to not to do what you don't want to do. That is inhibition of involuntary movements and performing the voluntary movements. So generally, the more common understanding is that we perform an action because we want to, but there are a lot of competing actions that the brain produces which we suppress every moment. So Huntington's disease is a disease in which some of these neurons which control the involuntary movements, like brakes for a vehicle, they die. So the body keeps moving erratically. It's called chorea, and chorea is defined as a non-rhythmic, involuntary, dancing-like movement. There is no rhythm. There is no pattern. So in tremors, you have a pattern. You have amplitude, frequency, etc. And for several other movements, you can define them by parameters, kinetic parameters. But chorea is something very erratic. And it's dancing like, and historically people call it as uh, it looks like someone who is drunk and moving the body parts without his or her control. So if you open the patient's brain after they die, you would see that the area which is controlling these movements called striatum is almost completely dead. So you will see an empty space there. And if you scan them when they are alive, you would see the same. But the extent of what you see depends on the severity of the disease. In the early stages, you might not see a significant loss of neurons, but in the advanced stage, you see that it's totally empty, the striatal region, basically.
Okay. So if I understand correctly, there are neurons in your brain that actually inhibit like involuntary movement.
Yes, yes.
Okay. And Huntington's is those neurons are missing, would you say? Or just
The,
not functioning?
no, the patients have them when they are born,
Mm-hmm.
but eventually they lose it.
Ah.
The disease kicks in. So when they are born, they don't have the disease, although they have the gene mutation carried in their cells, which is inherited from their parents, which I will talk about it a bit later, what kind of inheritance pattern and. So that is a very interesting question in the field. Why would someone get the disease after so many years, although they have the gene mutation from the beginning, since their birth? And there have been really, really wonderful research recently which is convincingly explaining us how this is happening.
I find it interesting. So I didn't know that there are neurons in the brain that actually inhibit movement because when people think of the brain, they think of like it's a computer, right? And it's making the moves in chess. But people don't think of it as preventing you from knocking the pieces over or doing something erratic. Um, why does the brain produce involuntary movement that needs to be prevented? Why does that even happen? I don't understand.
That's a great question. I'm not sure if I can really answer that, but it's a great observation that we actually not only do what we want to do, but we suppress what we do not want to do at every moment. Just imagine if this is also true for your thoughts. And what would happen if you lose the brakes for your thoughts, your judgments, your perception?
Hmm.
That's psychiatric illness. I'm kind of trying to tell you the conceptual framework and which neurons, which circuits, and how they interact. It's a bit more complex to discuss on a podcast. But why would brain have these unwanted things is a good question. Mm, um, I'm I'm not very sure, but I can tell you how it happens. So there are varieties of neurons, and some of these neurons are called tonic neurons. So tonicity means continuous activity. So they don't need a stimulus. They don't need a neuron behind them to stimulate this neuron to fire, right? So they continuously keep firing. And what happens if there is a firing of a neuron? The signal is going to the next neuron and the next neuron, and therefore it ends up in the muscle which acts, right?
Mm-hmm.
And the final act, which is the movement, is a summation or a sequential influence of one neuron on the succeeding neuron. So, if there is a neuron which is supposed to stimulate the next neuron, but it is not doing the job, then the next neuron is not stimulated. But what if there is a so again, there is another way to classify neurons. Some of the neurons are there to stimulate the other neurons, and some of them are there to inhibit neurons. So they are called inhibitory neurons. So imagine there are all these kinds of neurons, inhibitory and stimulatory, and a loss of inhibition will cause stimulation, right? And loss of stimulation is equivalent to inhibition. So like how computer works with the binary code, all there is to do in human brain is either to stimulate a neuron, the next neuron, basically, they communicate with this chemical stimulation, or not stimulate. And just imagine how wonderful it is that just this binary code of either stimulation or no stimulation can produce this wide variety of human experience, thought, movement. It's, I'm just giving you an idea of how complex this is.
Mm-hmm.
And I'm not a computer scientist how this happens in the computer, but the analogy is that even in the computer, it's just either 1 or 0. It's a binary code. Now, how do we generate this complex output? That is because there are millions of such neurons interacting in a network, and the ultimate neuron, which is going to the specific muscle or other area of brain, the output that receives from all the cumulative inputs will determine the final output. So there are very hierarchical levels of this complexity, wherein which a small alteration in one neuron can make a lot of changes in the network. So coming back to why brain should have this inhibitory neurons, why the brain should have tonic and clonic neurons. Tonic is like continuously firing, and the other neurons are non-tonic, which is they need a stimulation. So this is increasing the complexity of generating wide variety of outputs. First of all, there is a character of a neuron which is supposed to do a job or not to do a job. And then there is another complexity by the fact that they can continuously fire or they can only fire when there is an input. So that's another layer of complexity. And then the interactions between all this variety of neurons is another level of complexity. But the ultimate complexity is human cognition, consciousness, and perception. And how this all leads to that is even more a complex thing. So I think to add these layers of complexity, we have this, seemingly counterintuitive neuronal framework. Like, why should we have something to inhibit something we don't want? We have a neuron which is inhibiting the other neuron to not to do a bad job. If I'm sitting here and if my body is not able to hold up and I'm falling down due to my involuntary movements, those neurons have to be inhibited by some other neurons. But why in the first place my body should move is your question. Like, that's a fantastic question. But I can just tell you that it's because brain needed these layers of complexity, so it has different kinds of operational frameworks in terms of electrical activity and how they are arranged in sequence, inhibitory and stimulatory neurons. And maybe I don't know if it answered your question, but I really appreciate it's a wonderful question.
Yeah. No, it did. I just think it's so strange that like the whole concept of Huntington's, it's like it feels like it shouldn't even be like a disease or disorder from the standpoint of it's just using the input of the brain. It's like it's everything, the symptoms of Huntington's, like erratic movement. This movement is actually being ordered by your brain to do, basically. It's just not being inhibited. So it's very strange because it's not caused by external, like you said, it's a genetic thing. It's actually caused by a lack of a barrier, essentially, kind of. The brain is very complex. I'm really curious how we even discovered Huntington's. Like, how did someone figure out what was causing it in the first place?
Yeah. Before that, I wanted to appreciate your comment about how how Huntington's is a disease, although it's just doing what brain is commanding to do. Well, if you're doing something but you don't want to do consciously, then that goes back to your will, volition, consciousness. Is it good for you? No. But if you say brain is the seat of consciousness, and in the same brain you have unwanted firing, which is causing unwanted movements, why should it even exist? Really, really good thoughts, good ideas to discuss, and probably more complex to talk about in a podcast, but that's a great observation. And coming to Huntington's disease, it's a historical disease in the sense it has been reported for over 200 years. And in 1872, a doctor by name George Huntington, after whose name this disease is being named as Huntington's disease, made a first official publication describing a cohort of patients in Long Island, where his father, who was a physician, used to see these patients, and when he started practicing, he followed up their children. So it's a genetic disease, right? And then he made these observations and he wanted to tell world, this is a genetic disease and it's very complex. It's very interesting and devastating for patients. So he published his findings in a paper, um, and it was called Huntington's chorea. And when he described, he called it just chorea. Chorea is defining the involuntary movements. I'm not sure which language it comes from, Greek or Latin, but so he described it as involuntary dancing movements as the primary symptom, but they have all other different symptoms. And he beautifully described a lot of these symptoms, just being one person who observed so many patients, covering the cognition, psychiatric problems, bodily movements, and a lot of social implications. Like, I think there is a line which says, an adult, probably old man, having sexual behavior with younger people is abnormality, but why is this happening? Because like we have breaks for the movements, we have breaks for our thoughts to stop things that you shouldn't do in a social context, and that's governed by your frontal cortex. So I just simplified when I said it's only the movement part, but in Huntington's disease, lot more areas are also affected, like frontal cortex, but only in advanced disease. So not all patients have this disinhibition in the behavior, but some of these patients have. So yeah, so he described all these patients, and then there is like awareness about such an illness, and many more doctors and researchers noted about it. Fast forward in 1970s, let's say after 100 years, in the United States, we saw a revolution about Huntington's disease awareness, patients coming together and wanting to find a reason for why it happens. We only know that it's genetic, like it is passed on from parents to children, but how, what gene? So in 1870, when first it was described, there is no DNA concept. The DNA was discovered and the structure was only known after 1954, Watson and Crick's double helix model, and then subsequently we know the genetic code, and we knew what is a gene and then mutation. So Huntington disease actually, I would say, kick-started this movement in the scientific field of finding the reasons, genetic reasons for illnesses. If I am correct, Huntington's disease is the just second disease for which a single gene mutation was found as a causative factor. In 1970s or 60 to 70s, there were two prominent people who were affected by this illness. One is the pop singer Woody Guthrie, and his wife resolved to solve this, or to find a reason and solution for Huntington disease. And contemporarily, there was Wexler family. Nancy Wexler, whose mother died of Huntington's disease, was a psychologist at Harvard. If I'm correct, when her mother died, her father resolved to do something about it. And eventually, both of these people got in touch and they started this revolution. And they found through another doctor who published his findings, or who presented in a conference, that there is a place where there are a lot of these patients in a village. in South America, where people inbreed. They marry within the community, and they're so if you walk into this village, you would see a lot of patients with Huntington disease, like every second, third person in Venezuela. So, having known that, the team from the US went there. They sampled all these patients and their parents and their siblings. So you construct a pattern. Okay, mother affected, kids affected, and then kids of the kids affected. You construct this family tree and who is being affected. So by constructing these trees, you would know if there is a factor that is present in the patient, it should be present in the children of the patients who have the disease, but should be absent in the children who do not have the disease, right? So I'm just giving you the concept. And by then, there was no sophisticated genetic technology to just simply sequence the DNA, which we have like now. It's simple now, but then we didn't have. So there was a method to use bacterial enzymes, you know, which would cut human DNA into wide variety of fragments. And each of these bacterial enzymes have a tendency to cut at certain sequence, certain code. Human genetic code is made up of 4 letters, A, G, C, T. So let's say a bacteria wants to cut at a place where the sequence is A, T, T, C. And that is specific for that sequence. And that is the turning point in the whole revolution of molecular biology. So you take a unknown DNA sample, and then you put these enzymes and it keeps cutting. So you would know, when you arrange these fragments, you would know what will be the sequence, because you know that it can cut at a certain sequence. So they took all these samples. First, they constructed a family tree, and they, hypothesized that there should be a factor which should be present in all the patients and absent in all the healthy people within a family, because you can't really compare different people easily because there are many more factors playing around. And it's easy that you're following a same family tree. So when you put these enzymes, there are a lot of fragments generated, and there is a method to analyze the length of these fragments by running something called gel electrophoresis. So I'm just trying to simplify this, and when you run this electrophoresis, they found that patients had increased fragments, length of fragments, which are absent in the healthy people. Oh, then the causing factor is a length dependent. So someone has some extra code in the gene, and that discovery happened in 1983, which said that on chromosome 4, at a particular locus, there is something going on. There is some length increase in the genetic code in patients, but not in the healthy people. But we don't know what this is and what gene it is. So to describe a gene, we should have a structure. Gene has a structure, a promoter, coding sequence, non-coding sequence, which are arranged, alternatively, which are called exons and introns, and there is a structure. So by then, it was not easy. So from 1983, it took 10 more years to find what is actually this gene, and they named this gene as Huntington's gene, honoring the physician who discovered this disease, or told the world that this disease exists. And before that, even the disease was called Huntington's chorea, but after the gene was discovered, they named it as Huntington's disease, because we know the cause. Let's call it a disease, specific disease, than some loose term like Huntington's chorea, because there are other diseases which cause chorea. So to just differentiate, it's called Huntington's disease. So to sum up, by following these families where the illness was densely populated, like basically you should have multiple people. For example, if the patient has only one child, you wouldn't know, right? If the patient has at least one affected, one unaffected, you could do some comparisons. But what if you find some factors present in patients but not in the healthy person, but there are many such factors. So which one is causing Huntington disease?
Mm.
But if you have tons of such patients and children, then you can compare, you can see which is the common thing across all these patients and absent in all the healthy people, let's say. You know,
Mhm.
it can be vice versa. Absence of a gene can also cause disease, right? Absence of a certain sequence. So basically, this is a framework which is followed in the field of genetics to find the causes of certain diseases which are inherited from generation to the next generation.
Hmm. So essentially, they use process of elimination, kind of that general strategy, I guess,
Yeah,
to
kind of.
try to find the exact. Well, first they found the chromosome, and then they found the specific
Right,
genetic line in that chromosome 4.
right.
Right. Okay.
Awesome. Yeah, you got it. Yeah.
Okay. Um. So, and you said that it's extra code. There's extra code. There's the DNA sequence is longer in
Right.
people who have Huntington's, but so I guess two questions regarding that. Two questions regarding a cure, because so if you were to try to cure this disease,
Mm-hmm.
I guess how would you shorten, I guess, the length? Because my first thought would be to shorten it, but I'm curious if that's the strategy. And also, how would you cure the disease? Because you also said earlier a bit of a strange thing that people who are younger, they don't have it, but then they grow into it. Does that mean their DNA sequence grows longer, or just did you mean the symptoms grow more substantial as time moves on? And would that make it more complicated to cure?
Fantastic points, Bradley. I mean, these are the core ideas for which people worked for all these 30 years, you know. And you're right. And now, at least in last 3-4 years, it is very clear, and even they could model saying this could be the length increase which is causing the disease, and the speed of catching up with this length may be different in different patients, and you also have a starting length which is inherited, right?
Hmm.
So to answer your question, yes, there is an increase in the length. And previously there were two schools of thought, or predominant school of thought said it is not about length. I mean, length is there, and the disease is caused by a faulty protein. But this faulty protein is not effectively killing the neuron for a certain time. But as the disease progresses, the faulty protein starts accumulating and they form aggregates in cell. And cell generally, any cell has a scavenging system to clean out the garbage. Like we
Mm-hmm.
clean our houses to keep them clean. And these garbages of this faulty protein are so tough to be cleaned, and they start accumulating. And we have evidence because when you cut the brain, you stain and you see the Huntington protein is accumulating. Oh, then the simple answer is that protein might be accumulating over the years, and at one point the accumulation is so much, it hits a threshold that it's killing the neuron. And nobody thought earlier about whether the CAG. Okay, so I didn't give you the background. The mutation is caused by a CAG repeat expansion when it goes beyond certain limit, which is 35. So for normal people, all of us would have the CAG repeats, but within 20 repeats for most ethnicities. Actually, it is slightly different in the West and East and other ethnicities, but more or less it's 20. And what about 20 to 35? People don't talk a lot about it, but they found later that this 20 to 35, especially 27 to 35, these individuals will never develop disease, but the chance of these people giving a new mutation in their to their progeny is very high, which means the this bin of length 27 to 35 is highly unstable, and that would give rise to the disease causing length, which is 35 plus in the progeny. It's like a jump, significant jump. Well, this is an observation, and people are trying to find why this would happen only between this, length, but not, and all that sorts. So, um, for what was your question? I just lost in
Well,
details.
yeah, it was kind of, I asked like three questions in one, but it was about the cure.
Right, yeah. So now there is an evidence and convincing model to explain that the repeat expansions keep expanding, and then probably there is a threshold of this expansion which is killing. So the inherited repeats are over 35, and they can go on. I mean, the highest repeat reported was 200, which killed the patient within 2 years. Like, it's the highest. In my experience, I have seen mutations ranging from 36 till 113, and 113 repeat is so severe. I mean, so predominant, so toxic that the disease occurs very early in life, which means repeat length has a correlation with the age at onset. If you have longer repeat, the age at onset is less. Now, this is one fact people observed by looking at their mutation and their age at onset. If you make a graph, it is showing the thing. Now, the actual length which is killing is different in the brain, and we don't know. We only know the repeat in the blood, which is being passed on to them from their parents. But what exactly is happening in the brain, we do not know. We can only know when they die, right? Interestingly, your repeat length, which is being inherited from your parent, is still explaining the age at onset with 60% accuracy. So there are other factors which are affecting your age at onset. For example, if you take two patients with same repeat number, 42 and 42, they do not have exactly same age at onset. But if you take a large set of patients and you plot this graph, correlation graph, you would see that it's fairly showing that there is a relation, and it is explaining about 60% of variation in age at onset is caused by the inherited mutation length. What about the other 30-40%? To know this, what people do is they take all these patients and they sequence their CAG repeats, and they also sequence many other genes, almost all the genes in the human genome, which is called genome-wide association studies. And then they see that which of these mutations are associated with earlier age at onset or late age at onset, which means these are the factors which are affecting the disease onset beyond the effects of CAG repeats. Right, that's one aspect. And yeah, for your question, yes, the increase in length. Now, to cure, when you said, the first thought that came to your mind is to make this repeat less, make this repeat small, repeat length small, which is perfect. Theoretically, that's a beautiful idea. How do you do that? So back then, definitely nobody would have thought, how can we even, constrict? And the major problem for genetic illnesses is that you have this gene in every cell of your body. It's not like it's there only in your finger. You can repair it somehow. But because you got it from your parent, it's in every cell of your body. How do you repair every cell? Right?
Mm-hmm.
But we only need to repair at least brain cells to cure the disease, which is decent. But even in the brain cells, how would you cure all these thousands and hundreds of thousands of neurons? How do you enter into this cell, and then you. Now in 2024, we are in 2024, we have technologies to do that. Theoretically, in cells, if you take cells in a dish in the laboratory, we have technologies, a beautiful technology called CRISPR, for which the scientist who discovered this was awarded Nobel Prize a couple of years ago. This technology uses certain enzymes which go and cut the genes where you want. You can design in a way that it goes and cuts at a certain specific site in the genome. So if you design these probes in such a way that it cuts on either side of CAG, you would remove the CAG. You can remove an entire gene. All these things can happen. For this technology to actually translate into human therapeutic avenue, there are a lot of hurdles, and research is going on. But there were many other approaches which people thought of can cure the disease. Number one, the protein is accumulating, and there is strong evidence, and people strongly believe it, that these aggregates are causing the cell death. How about stopping the aggregation? People tried certain things in the laboratory. Some worked, some didn't, and nothing actually worked in the patients. Second, what about stopping the protein from forming? What about stopping the gene to be expressed? So a gene generally becomes something called an RNA, which is a messenger RNA, which carries the message. And this messenger RNA is the template on which ribosomes, cellular particles, molecules, will synthesize the protein,
Yeah.
So how about not letting this protein form? There are two problems with this. First, how do you do? And second, there must be some good function for this protein, right? If evolution has given you this protein, there must be some function. What would happen if you stop the function of this protein? It might have some deleterious effects. So anyway, people went ahead and they designed certain molecules which would go and bind to the mRNA. After gene makes the mRNA, they would go and bind to the mRNA and destroy it, so that the ribosomes cannot make this protein from the mRNA. And this is the most talked about, tested in the last 10 years. And in the laboratory where I work, we test a lot of these molecules. They are called antisense oligonucleotides. They're called antisense because they have the complementary sequence which will go and bind to this mRNA, and then they trigger their destruction. So the first human trial, which gave a lot of hope, in 2017, there was a lot of hope and positive, news about the cure because of some favorable findings in a trial where they used these ASOs to destroy the mRNA, not letting the protein form in human patients, by injecting these drugs into the spinal cord, which will eventually go to the brain, through a fluid called cerebrospinal fluid. What they observed is that it's definitely decreasing the protein. So boom, it worked. But these ASOs were non-specific. So coming back to what actually a genetic disease is, genetic disease is caused by a mutation, and you have 2 copies of every gene in your body, one from your mother and one from your father. For some diseases, you need to have 2 faulty genes for the disease to occur. They are called recessive illnesses, recessive genetic diseases. And For other diseases, you only need one copy. Even though other copy is healthy, the unhealthy copy can override the effects of healthy copy. That's the reason they are called dominant diseases. And the former one which I mentioned are called recessive, because the healthy protein is the dominant there. The mutation is recessive, so it cannot override. Huntington's disease is a dominant disease, dominant mutation. So one copy is actually lethal enough to cause the disease. So if you really stop this faulty protein to form, but not stopping the healthy protein, we are good. At least half of the protein is there. I mean, half the amount technically, at least. That's not a straight correlation. Maybe body can produce as much it wants with one copy. But the first AOSOs were non-specific. They would block both the proteins. So they were designed in such a way that they would block both the mRNAs, faulty one and the normal one. So the trial apparently looked that it's working because the Huntington protein was decreased. Eventually showed that there was no improvement in patient symptoms, and it even worsened. That was a bit shocking, and the clinical trials were stopped. So eventually people got interested in why blocking normal protein causes the disease, worsens the disease, which means normal protein has important functions even in the adults. Because earlier people thought Huntington disease probably is an important protein, but maybe after an age it's not very required or something like that. But by these findings, people wanted to know what is the actual function of this protein and what are we losing by blocking it. So one of the goals of my lab, my supervisor, Jeff Carroll, is very interested to understand what is the actual function of this Huntington protein and what is happening when you block it. I mean, it's worsening the disease is what we found, but why? How? What are its functions which are blocked for the disease to get worsened? So our lab has these two goals: to test some molecules, and so once these trial results came, people started working on developing specific ASOs which would only prevent the faulty protein to form, not the healthy ones. So these are upcoming trials, and there's a lot of hope. And again, coming back to how you deliver and how frequently you have to dose. Let's say it's an injection that you have to give to your spinal cord. It's not good to give every few months. It's not easy, and it's expensive. And there are risks of putting an injection in your spinal cord. So people are working on the chemistry, how to block it and how to deliver it, all these kinds. So shortening CAG is a very theoretically beautiful concept. And there is some research and which is not yet published from a group in Canada, a scientist in Canada. They found a small molecule, it's a chemical compound, which actually showed that when you treat human cells with this compound, the length is shortening. So even that is not yet closed. So there are beautiful things happening, but Huntington disease is a prototype of how we are challenged as human beings, although we have so much, intelligence, technology, and theoretically, like you said, we can shorten the CAG, we can block the CAG, we know all the things. But we could never succeed to get something into the clinics as a cure, although we knew the mutation since 1992, which is like 30 years, more than 30 years. So I always feel fascinated, challenged, all kinds of emotions. Maybe it's a prototype, and there are many such illnesses, to be honest, but Huntington disease happened to be one of the earliest diseases to be researched and a cause was found. There are many such diseases, actually speaking. You know, that's about cure.
Hmm. I'm curious, why was it not tested on animals first? Because I know usually a lot of times with like viruses and um, illnesses that they'll test on an animal first. Is this, since it's a genetic disease, is it only unique to humans or?
Great question. So, I mean, we don't know. We couldn't sequence all the animals in the planet. We
Mm-hmm.
even if we had sequenced, let's say, monkey, we don't sequence all the monkeys, right? So we don't know if naturally Huntington's disease exists in any species. We have no evidence yet. Probably not. I don't know. But we can produce an animal with human disease. How do you do that? By inserting this mutation in their Huntington's gene. That's called knock-in. You're knocking in certain code. Or you can produce a transgenic animal where you take the entire human Huntington's gene and put it into the animal, which is not so natural because you are taking a foreign gene totally and putting using technology to insert somewhere in the genome, and it starts producing this mostly overexpressed gene, because when you put the gene you also put the promoter. Promoter is something that drives the expression of gene. Without promoter, gene is not expressed, so you have to put human promoter as well. Not a natural state of expression. So that was the first model, and then improvised models are knock-in models. So they found that mouse also has a Huntington gene like humans. And lot of homology, like homology as in if you align the sequences and compare, a large part of it is same. It's, little different, but not largely different, largely same. And the repeats, CAG repeats in the Huntington gene of mouse were very less, about 7, which is also fascinating. Maybe evolution actually increases these repeats. From mice to humans, we see this increase. So there must be a purpose. And this purpose has gone wrong beyond a limit. We don't know. And there were scientists who were very interested to understand the evolutionary aspects of Huntington gene and its repeats. And it's beautiful that you find a Huntington gene-like genes starting from amoeba. It's a protozoa, unicellular animal, unicellular creature, and then almost all of the species have Huntington gene in different levels of homology with human beings, but the mice one is very close. So you can produce Huntington disease by increasing the repeats. So if you take this Huntington gene of the mouse, which has 7 repeats, and I increase it to 100, and the mouse shows the symptoms. There you go. And now you use this mouse to understand the mechanisms, how the neurons are dying, like how the aggregates are forming. And we are limited by the availability of human brains. We can only get when the patients die and they donate. But with animals, we can produce them, we can control their behavior, so we can put both animal with Huntington expansion and without a Huntington expansion in a controlled environment, give same food, same clothing, and all that. So it's easy to compare when the conditions are controlled. Whatever changes you see are because of the mutation, because everything else is same. But in human beings, we have so many attributes, we have so many contexts. We have socio-economic disparity, educational disparity, nutritional disparity, and also a lot of other genes, right? In the background, like I expressed. In genome-wide association studies, they find the other modifiers of this disease. But in mice, you take same mice and you serially reproduce them, breed them, and they have mostly same background, right? And then you make a mutation. They're called isogenic animals. And all the drugs will be first tested in animals before human beings. Recently, there is some, change in these protocols, because, like you see, when COVID happened, we needed a drug so fast. There's no time to really test, you know.
Right.
Even in Huntington and other rare genetic diseases where they are so fatal, not even Huntington, but there are diseases which kill the patient within first 5 years of life, first 2 years of life. So they're anyway going to die. Instead of waiting for the drug to pass all the screenings from animals, basically they do a mice trial typically, and then a primate, monkey, or then human. It's long. So FDA, which is the regulatory authority of controlling the trials, has been little flexible. I mean, someone has to make a proposal. I want to try this drug because the patient anyway dies, and it's so pathetic, and you can't wait. You know, so and patients are willing to come forward. The parents are willing to do this. So there has been a change, but generally speaking, it was tested. So for your question, was that effect which we saw in human beings was seen in the animals before, right? I think no. That's the reason they went ahead. And now the question comes, how good are these models? Because we didn't see this in animal, but in the human being. So that is a very debatable thing. And now we have technology where I take your cells, your scrapings of your skin, and make neurons out of them. So those are your neurons. So I take the drug and test in your neurons, which is a robust evidence. Again, it's not perfect because it's outside your body. So even that is not perfect. But you can complement these two, where you can take an animal which is a living system, and we can take your own cells which are specific to you but not in a living system in a dish. So you can compare and complement. And this beautiful technology with which you can develop your any kind of human cells or tissues from any cells, any other cells, is called induced pluripotent stem cell technology, for which again Nobel Prize was awarded a couple of years ago. Beautiful. So beautiful thought someone got when they thought about what makes an embryo, a zygote, grow into a fetus and adult, right? How to can we reverse this process and make an eye cell again back in its lineage towards zygote. So there is something called stemness. Stemness is the ability to divide into multiple types. So gradually, zygote is the only cell which can divide into any cell, which is called totipotency. And then zygote becomes different other cell types. They are pluripotent, which means they can divide into several other cell types, but not every human cell type. And then pluripotent becomes multipotent, and so you lose the stemness. So scientists discovered a way to reverse engineer this and reach to a stage where you can go to that level and start differentiating into a different daughter cell from the same lineage. So let us say neuron and your skin cell shares the same mother. So you make the skin cell into the mother and then make it into a neuron. It's a beautiful thing. So what I'm trying to say is, like, we have so many technologies being developed which are trying to fill the gaps within the models. So there is hope, and yeah, it's been tested, but yeah, it wasn't useful.
Wow. Very interesting.
Yeah.
Um, if you were able to find a cure for Huntington's disease, how revolutionary do you think that would be for other brain diseases? Like if we find a cure to Huntington's, would it unlock a lot of different things, or is Huntington's so unique that finding a cure for Huntington's is like it would just be that basically?
Fantastic question, and that's another thing. So there are about 30, I'm not very sure, it could be more, human diseases all of them affecting brain and sometimes brain plus some other organs, which are caused by CAG repeat expansion. So if you find a solution for Huntington which is addressing the repeat length, let's say decreasing the repeat length, blocking the repeat length, well, you can solve all these diseases theoretically. I mean.
Hmm.
So, yeah.
Ah, fascinating.
Yep.
Another thing I wanted to ask is something you mentioned before. Um, you mentioned that Huntington's was
Right.
a dominant disease. Okay, so I'm having a hard time wrapping my mind around that because um, I feel that diseases should technically be recessive because, well, if you go off evolution, right, the and you have two chromosomes of each, then the body should want to favor the one that would increase your survivability. Let's say, like through evolution, it makes sense why diseases would be recessive. Um, Perhaps this is kind of a silly question,
No, not at all.
but
Uh,
why is it dominant? Why?
it's not silly, it is a very profound question. And the terms dominance and recessive are labeled by us. It's
Mhm.
more complex than,
Mhm.
we are simplifying to say, okay, presence of one copy is adequate to cause the disease, and presence of two copies is mandatory cause of disease. So, it's a kind of labeling. But there's much more to it. So there is something called gain of function and loss of function. So if a mutation causes the gene to produce a protein which is not functioning at all, so there is a protein which is supposed to do something, let's say catalyze a reaction, and it's not doing it, it's a loss of function. Now when this gene is not functioning, its sister copy, the homologue from the other parent, is still working.
Mhm.
So most of the genetic diseases which are recessive are loss of function mutations. Naturally, understandable, right? Again, it's not as simple. Again, there are exceptions, but it's intuitive to understand that there is one copy which is not working, but the other copy can compensate. At least half compensation, worst case, right? But what if the mutation is giving it a new function? And this new function is toxic, it's lethal. So having the other copy functional doesn't help, because the mutation is creating a new function, new effect, new interaction. This is called a gain of function. And a majority, almost all, dominant diseases are gain of function. Makes sense? Right. So at the broader level, there are exceptions, but yeah, it's too much for us to discuss now. But yeah, that makes sense. Yeah.
Mhm. Hmm.
Does it answer your question, or is this more?
Yeah, it does. Also, another thing that was going through my head was if a disease is dominant, then I know those are words that we put
Right.
onto the disease, but shouldn't more people have the disease? Like, shouldn't
Right.
everyone have it?
Right. Right.
Well, I guess technically everyone probably does have it, right? Because everyone has this strand of genetic code, right?
Mm-hmm.
But some people have it longer than others. But I remember you mentioned that if someone has like 27 to 35 uh, length, a strand of this Huntington's
Right.
code,
Right.
the chance that their offspring will have 35 or more is higher, right? So shouldn't a lot of people have this disease? Or are you able to, I guess, explain why?
Right.
Is
So
it classified as a rare disease or?
at the moment, yes,
Okay.
but who knows?
Yeah.
But yeah. So what affects the prevalence of a disease, especially genetic disease, uh, is based on the lethality of the mutation. I mean, if it's too lethal, the individual with the disease wouldn't even pass it because he wouldn't attain the reproductive age. And the mode of transfer, like dominant and recessive. So in case of dominant, like you said, it's an intuitive thing that more number of people should be affected. And in recessive, since you need 2 copies, less number should be affected. You know, the fascinating thing is it's the opposite. Because in dominant diseases, they are so dominant that they kill the patient before they attain reproductive age. Again, this is subjected to all these conditions. If a dominant disease is not so lethal, dominance does not always mean that it's so lethal, but dominant only means, in the context of describing a genetic disease as dominant, is 1 copy is sufficient, is overriding the other copy. But if it is also happens to be a very strong disease which kills the patient, the chance of spreading it is less, right? In the recessive thing, although you need 2 copies, the person with 1 copy is healthy, doesn't show any symptoms. The chance of him reproducing is 100% like you and me. And eventually, over the years, the people with 1 copy are so much, like, you know, and yeah, I mean, it's too much into weeds. I don't think we should go, but the allelic frequency, allele is the variant of the gene, so, you know, they call it alleles. The allelic frequencies in a population of the recessive diseases will be much more than the dominant, because they are not killing the patient, number one, and they're reproducing, and eventually, over the time, the prevalence of these alleles will increase. Again, when the 2 copies of the gene are present, those diseases could be even worse than the dominant disease. It depends on the protein of interest, you know. So many of these enzyme disorders which are recessive, they're pediatric, like the onset is at young age and they don't reproduce. So nature has a way of keeping a check of, I mean, it's a story that we tell, it's nature's plan or whatever, but it's a dynamic which needs to be in equilibrium. So there are concepts in genetic, how the equilibrium is attained and maintained, and what can disrupt the equilibrium. Let's say half of this population migrated, some new people came and they had a different mutation, whatever, you know. So a lot of factors affect the prevalence of diseases and the time. But I want you to note one important thing. This Huntington disease or its similar illnesses which are dominant but are late onset or middle age onset, they pose a very important problem, because you do not know that you carry the disease. You would eventually, you would not think about not marrying or not reproducing, so you would end up passing this gene and later discover that you passed it and your children are at risk. But the other point also is that since they are strongly genetic, if you do a genetic testing, even for the infant, you will know whether he or she is going to get the disease after certain age. That creates another problem, whether you should test infants, whether you should test kids or not, because you are revealing something to them which is very strong, which is very life-changing for the individual. It could be positive or negative, and in many cases it's a negative, because how do you deal with such an information? I mean, the only positive thing is you plan your life ahead, you know, but you know, the common observation is that it really breaks that person to know that they have the disease, and they waste or they involve in self-destructive behaviors or even commit suicides even years ahead of their predicted age at onset. So it's a tricky thing. But the point I was trying to say is, since they are of late onset, the reproducibility is very high, and since they are dominant, the occurrence of disease is very high with less frequency of allele prevalence in the less frequency, less prevalence of the allele in the population compared to the recessive will be high because one copy is causing the disease. We don't need the two copies. And then also they are reproducing. Now, if you take this phenomenon over the years, I didn't tell you one point. I told you this, but I didn't emphasize that more the repeat length, earlier the disease. But there is also one point, it's called anticipation, which means from one generation to the next, the likelihood of the increase in this expansion is very high. So children always have younger, not always, most instances. It's not a hard and fast, rule, but mostly children have longer repeats and earlier onset. And imagine over the years, it's getting longer, and then it's the survivability is becoming less and reproducibility is less. So most patients do not attain the reproductive age, then it does not pass. So it's in an equilibrium again.
I see.
But it takes years and years, hundreds or thousands, we don't know. But I'm saying it's a dynamic thing. Yeah.
How close do you think we are to a cure? Like, do you think in the next 10 years we could have a cure?
Yeah, great question. I probably am not enough qualified to make a real prediction, but I've been in this field and, kind of have a rough idea of where it's going. Every year we see a development, a new technology, a new way of delivering the drug, new chemical compound, which is so promising. If you ask me personally, it's a belief I cannot substantiate, because how can you be sure that 30 years of things failed and, are you expecting a cure in next 5 years, 10 years? Also, what is cure? That's another question I want to ask you. But before that, I have a strong feeling and strong hope that in 5 years there will be substantial improvements in Huntington disease therapeutics. Now, what is cure is something we need to talk about. What do you mean by cure?
Well, I guess the first thing that I think of when I think of cure is reduction of the involuntary movements or involuntary thoughts. I don't know if you can completely remove those, because probably everyone has a little bit, you twitch or something every now and then. But that seems like, I don't know a lot about the disease, right? But that seems like the first step to a cure.
So what you referred to is a reduction in symptoms.
Right.
But what if there is a way to reduce the symptom but not stopping your disease? Do you think it's a cure or temporary treatment?
That's a great point. What do you think?
We do have these things.
We do?
Yeah. So we have drugs which... So like I said, again, going back to how neurons work, neurons release chemicals which release the next neuron. And some of these chemicals are inhibitory, some of these are stimulatory based on the neuron type. And some of these neurons are continuously firing, some are... So all this stuff. Now, we have a way, for example, loss of brake is causing involuntary movement. Means at one point, some neuron is firing, which is not supposed to be firing, and ultimately leading to the stimulation or excitation of the muscle. So there are multiple points actually you can target, but there are drugs which will, let's say dopamine is, the chemical which is believed to cause chorea. In the sense everyone has dopamine, but these patients have dopamine in an area which would cause the involuntary movements. I mean, the same area might have dopamine in all the individuals, but because of the absence of some other neurons which are interacting with this dopamine, you're getting the disease, right? So how about blocking the dopamine? So dopamine is doing something, and the other neuron is antagonizing. Now you don't have this. That's why dopamine is exerting its dominant effect, and you're getting the movements. What about blocking the dopamine? Well, that works temporarily. Why would it won't work eventually? So these actually go back to the same layers of complexity of neurons and how binary code is generating. But essentially, I'm trying to tell you that without stopping the disease progression means not stopping the toxicity to the neurons. We can control the movements temporarily. And at one point, they don't work anymore. But they kill the neurons equally as the patients who is not getting treated. So eventually they are same, right? So reducing symptoms is very much there. And most patients respond to different extents. It's not like 100%. I can't 100% remove your chorea. 50% is decent enough. You're not falling down from the sitting position from chair, which is good, which is better than falling down, right? You're not dropping things from your hands, which is a great thing. But when we talk about cure in Huntington's, general perception is that you remove the cause of the disease so that it doesn't occur. But that, I think, is also other extreme of thinking about a cure, because you know for a fact that you can't really remove this mutation from all the neurons. Even if you are blocking the formation of protein, what if you miss one injection of these molecules which are blocking the mRNA? So you still get symptoms. So how I look personally, and I think my supervisor, he also mentioned it somewhere, which is what the belief I also share is that if your quality of life is substantially improved, if your lifespan is improved by, let's say, 10 years, boom, it's a cure. I mean, with confidence, if I can say that this molecule can increase your lifespan by 10 years, it's significant improvement in therapeutics. That's what I mean, yeah.
Okay. Nikhil, it's been great talking with you, and thank you for your time today.
My pleasure, Bradley, and thanks for having me. I've never done a podcast, but it felt very good. You have asked a wonderful, critical questions in the field, so I'm very happy.
Oh, thank you.
Yep.
Hopefully, we'll talk again soon.
Yeah,
All right,
look
bye.
forward to. Yep.
What do you think?
I was giving a long responses because